Okay, so you scrape your knee while riding a bike, or you cut yourself while cooking with a knife, and it bleeds. Yeah, but after like a few minutes, the bleeding stops. In a few days, has started to heal and is healed. But in a year, it's almost as if nothing ever happened. You are a full-fledged multicellular organism with functioning systems and organs, but you were once a unicellular fertilized egg. Let's talk about it. Hi guys, good day, depending on when you'll be watching this, and welcome back to the channel. On today's episode, we are going to be talking about cellular reproduction. So as the name implies, cellular reproduction is how cells replicate themselves in order to continue their lineages. By definition, cellular reproduction is when a cell known as a parent cell replicates its DNA to form two or more daughter cells, which are the resulting cells from that DNA replication. And it's very important for a bunch of reasons, some which I already stated in the introduction to this video. It's important for growth and development. It's important for regeneration and repair. So when you hurt yourself, you're able to heal. In some organisms like the zebrafish and in some types of lizards, they can actually regenerate full organs like a fin or a tail. That process is brought about by cellular reproduction. And it's also important for reproduction because cellular reproduction is the process that brings about the gametes, which are the human sex cells, sperm and egg. Let's assume that you're already familiar with cell theory. If you're not, I'm just going to tell you what it is. So basically, cell theory is a foundational principle in biology which states three things. First of all, is that all living things are made up of cells. Yup, plants, animals, human beings, everything is made up of cells. And the second one is that the cell is the basic structural and functional unit of life. You can't really find life beyond the cellular level. It doesn't work like that. And the third one, which actually gives context into our discussion today, is that all cells came from pre-existing cells. So these three postulates are what we call the cell theory. And the last part of the cell theory is what we're going to explain today, how cells actually emerge from pre-existing cells. So there are two major forms of cellular reproduction, namely mitosis and meiosis. Mitosis is essentially a cell duplicating itself. It is when a parent cell forms two daughter cells that are genetically identical to the original. Meiosis, on the other hand, is when a parent cell brings about four daughter cells, and those cells are actually haploid. Like I mentioned earlier, cellular reproduction leads to the production of gametes, and is this process called meiosis that actually brings about the haploid cells that are our sex cells. For the purpose of this video, we're going to be discussing strictly mitosis. If you came for the whole shebang, sorry. We're talking about only mitosis. Mitosis is very important. Remember all those importances and functions that I listed earlier on in this video. Mitosis actually is responsible for them. Repair and regeneration of damaged cells. Mitosis is responsible. That's why when you cut yourself, you're able to heal. The process of growth and development, how we go from a fertilized egg, zygote, to an embryo, to a fetus, and to a baby. That whole process of growth and development is brought about by mitosis. Mitosis happens in stages, right? But before I explain the mitotic stages, I think it's important for you to get familiar with the cell cycle. What is the cell cycle? As the name implies, the cell cycle is a sequence or series of events that encapsulate what happens when a cell grows and replicates. Scientists have been able to identify distinct events that happen in the cell cycle. The cell cycle is divided into two major points: interphase and M phase, or mitotic phase. These two phases are the major ones. They also have minor subphases, but let us start with the major ones: interphase and M phase. M phase is the mitotic phase, and that is when a cell undergoes mitosis, as the name implies. So it's basically the whole mitotic process. However, just before and just after this mitosis phase, we have what we call the interphase. We've grouped all the processes that happen before and after mitosis into just this phase called interphase. So if you're a bit confused, I'm going to use this as an example. I want you to imagine that the cell cycle is indeed a cycle, like a circle, like this, right? So let us say that this is the entire cycle, and then just from here to here, this short distance of the cell cycle is the M phase, during which mitosis actually occurs, right? So as you can see, the area just before and the area after mitosis is actually the same area. So starting with interphase, interphase has three subphases: G1, S, and G2. G1 is a period of growth just after mitosis, during which the cell has to reach its full functioning capacity. When a cell is formed, it's kind of like just a baby. It resembles the parent cell genetically, but it still has to grow up to a certain point in order to become a mature cell. So this is what G1 is about. In the S phase, this is where DNA replication occurs. The cell duplicates its DNA into what we now call sister chromatids. So each chromosome with its respective duplicates, they are sister chromatids. These sister chromatids are actually tightly adhered to each other along their length by what we call cohesins. They're held together at this point, but they'll be released later. We'll see later on in the mitotic phase when these cohesins are removed. In animal cells that have centrosomes, the centrioles are duplicated and two centrosomes are formed. And this is important because it's kind of like the early stages of the formation of the mitotic spindle, which we will talk about later. So after DNA has replicated, we move over to G2. So G2 is just a brief period of growth during which the cell kind of synthesizes protein, the equipment that it needs to undergo mitosis in the M phase. And after G2 is complete, then the M phase can begin. The M phase is characterized by 5 subphases. We have the prophase, the prometaphase, metaphase, the anaphase, and the telophase. Starting with the first one, the prophase. So in the prophase, the sister chromatids condense and become more tightly packed. This is important because it reduces both its volume and length. A condensed chromosome is about 7 times less volume than the original and about 1,000 times less in length than the original. This is important because it kind of fortifies the DNA. As it moves around the cell during mitosis, it prevents DNA breakage. At this point, the cohesins that hold the sister chromatids together are released except for the cohesins at a certain point called the centromere. This condensed chromosome form with the centromere, which is that point where the sister chromatids are seen to be held tightly together, is actually what we see under the light microscope. Chromosome is too little to be seen when it is elongated. When it condenses, that is when we're able to observe it, and then the centromere is observed as a tight, you've probably seen all those diagrams, as a tight constriction at some point along the sister chromatids. Most of the pictures that we see depict the centromere in the middle of the sister chromatids. However, the centromere is actually chromosome specific. Different chromosomes have a centromere at different points along the sister chromatid length. During this phase as well, in animals, the centrosomes are duplicated, begin to move away from each other in order to start forming the mitotic spindle. The centrosome is basically a center of radiating microtubules that radiates throughout the cell in every direction. When the centrosome duplicates and moves away from each other, they form the mitotic spindle, which is 2 centers of radiating microtubules forming at opposite sides of the cell and overlapping at some point in the middle. In plant cells, there's no centrosome, no centrioles. You still see a mitotic spindle formation, but instead of centrioles at the center, we have lines of cross-linked microtubules. Next, we have the prometaphase. During this phase, the nuclear envelope breaks down and the nuclear core proteins are disassembled. The phospholipid bilayer that makes up the nuclear membrane absorbs into the endoplasmic reticulum. So this allows more room for the mitotic spindle to form and allows the chromosomes to enter the cytosol to perform the mitosis. Pause. I think it's important to note at this point that the process I have been describing is actually known as open mitosis. Some eukaryotic cells undergo another form of mitosis known as closed mitosis, during which the mitotic spindle actually forms inside the nucleus. Scientists believe that open mitosis evolved as a result of chromosomes being inherently more large than the unicellular organisms that closed mitosis evolved from. So they needed more space, and so the nuclear envelope needed to be broken in order to allow mitosis of that volume of DNA. Some organisms also even have a hybrid of open and closed mitosis with varying degrees of nuclear inclusion, but yeah, you can probably go read up on that somewhere. So during this phase as well, there's a protein complex known as kinetochore that forms at the centromere of each sister chromatid, and then a structure known as kinetochore microtubules, which emanates from each point of the mitotic spindle attached to the kinetochore of each sister chromatid. And this is important because this is exactly how cellular reproduction takes place. This is how the chromatids are shared and distributed equally amongst the 2 daughter cells. There are also non-kinetochore microtubules that extend from each spindle pole, and those kind of overlap in the middle, not attaching to any kinetochore or chromosome. The next phase is called the metaphase, and during this point, the mitotic spindle has reached its full maturity, and the kinetochore microtubules align each sister chromatid pair at the center of the mitotic spindle, which is also known as the metaphase plate or the mitotic spindle midpoint. So they bring all the sister chromatid pairs into alignment, and then mitosis is ready to start. The requirement for mitosis to begin is that all sister chromatid pairs need to be at the center at the metaphase plate, with each sister chromatid needing to be attached by kinetochore and kinetochore microtubules to either the left or the right side of the cell. And when this happens, mitosis can begin. The next phase is the anaphase, which is actually the separation. This is where the separation happens. An enzyme known as separase now then cleaves remaining cohesins left at the centromere of the sister chromatids. And when these cohesins are cleaved, sister chromatids are allowed to move to separate opposing ends of the mitotic spindle, and this is the moment at which chromosome segregation actually happens, and the chromosomes that were known as sister chromatids are then known as daughter chromosomes. This phase and the phases leading up to it are really important because I feel like I've just explained how cells consistently produce genetically identical daughter cells. These processes are important: the alignment, the kinetochore microtubule attachment. These are mechanisms that are put in place to ensure that each cell is a genetically identical copy of its original. The next phase is basically the winding down phase, is the telophase, and during this phase, the mitotic spindle disassembles, and then the daughter chromosomes then decondense into their original length. This decondensation triggers RNA transcription, and then a new nuclear envelope forms around the chromosomes by the extension of the endoplasmic reticulum into which the initial nuclear envelope had been absorbed, and then the nuclear core proteins reassemble, forming two identical nuclei or two identical daughter cells. What typically happens after this process is cytokinesis, which is the division of the cell cytoplasm to form two physically distinct cells. And cytokinesis happens in different ways depending on the type of cell, whether it's a plant or animal cell, and you can go read up about that. That basically encapsulates the major events of mitosis. This is just a summary, as you can tell from the description. There are still so many things that you may need to know and you may want to read about, including the actual mechanism involved in moving a sister chromatid to one end of the mitotic spindle. That is how the kinetochore microtubule is able to drag the chromosome. It happens by two mechanisms, but I'm going to leave you guys with that as food for thought to go read up about mitosis in plants, because I majorly just explained what happens in animals. Plants don't have centrosomes, so their mitotic spindle formation would be kind of different. You may also want to read about how these processes and this cell cycle is regulated, because obviously many things could go wrong, so our body kind of has mechanisms that ensure that each stage of the cell cycle goes according to plan. And actually, dysregulation of this cell cycle is actually what causes cancer. When cells don't know when to replicate their DNA, or when to start or stop growing, or when to divide, that is what causes the different types of cancer that we see. So you may want to do some more research into this because it's really interesting. This is just a general summary of what typically happens in the cells that most people talk about and your professors may want you to be familiar with. Thank you guys for staying to the end of this video. It's my pleasure to explain some of this stuff to you, and see you on the next episode.